Thank you for attending my talk. The title of the presentation is Focus Ultrasound-Induced Blood-Brain Barrier Opening and Systemic Anti-CD40 Agonist Enhances Monocyte Infiltration of Intracranial Melanoma. I confirm that I've not got any conflicts to declare. Melanoma is an aggressive form of skin cancer that has a high tendency to spread to the brain. When it does, it can have devastating consequences for patients, including loss of independence, cognitive dysfunction, and functional impairments, as well as death. Immunotherapy, specifically immune checkpoint inhibitors, is an effective treatment against melanoma, but we need more tools to tackle this disease, and melanoma brain metastasis in particular. Anti-CD40 agonists are exciting agents that have diverse immunomodulatory effects. The primary mechanism of action against cancer is through activation of dendritic cells and stimulation of a cytotoxic T-cell response. Therefore, using a two-site model of melanoma brain metastasis, we sought to investigate whether focused ultrasound blood-brain barrier opening could enhance the delivery of a systemically administered anti-CD40 agonist to the intracranial tumor, and therefore improve control of this disease. The experimental timelines involved a sequential inoculation of overexpressing B16F1 murine melanoma cells, first in the flank, followed by intracranial injection. Then, the Black 6 mouse was treated with either an anti-CD40 agonist or a non-targeting antibody. Finally, the treatment was repeated, but this time following MRI-guided blood-brain barrier opening induced by FUS. Interestingly, we found that treatment with anti-CD40 with or without blood-brain barrier opening did not improve survival, and all animals died as a result of their intracranial tumor. Interrogating the peripheral immune profile of tumor-bearing animals that received either anti-CD40 or IgG revealed quite marked changes in the circulating immune cell populations. In particular, there was a dramatic reduction in circulating B cells. However, the myeloid compartment was also prominently affected, with an increase in circulating monocytes in the anti-CD40 treated group compared to the IgG group. Delving into the effects on the immune population in the intracranial tumor, treatment with anti-CD40 agonists resulted in an increase in infiltration of monocyte-derived dendritic cells compared to animals receiving IgG. This was not significantly impacted by the application of FUS-induced blood-brain barrier opening. Correspondingly, there was also a loss of XCR1 expressing conventional dendritic cells in the intracranial tumor following anti-CD40 agonist therapy. Furthermore, there was increased infiltration of inflammatory monocytes into the intracranial tumor following anti-CD40 agonist and FUS-induced blood-brain barrier opening. This was significantly greater than in either the anti-CD40 group without FUS or the IgG group with FUS. Conversely, there was no significant difference in CD8 T-cell infiltration in the intracranial tumor between any of the groups. So, to conclude, we know that anti-CD40 agonists have diverse roles in regulating the anti-tumor immune activation. Surprisingly, we found that anti-CD40 alone and in combination with FUS-induced blood-brain barrier opening did not improve survival in a two-site model of brain metastasis. However, it was evident that systemically delivered anti-CD40 did stimulate a peripheral immune response, predominantly in the myeloid compartment. When combined with FUS-induced blood-brain barrier opening, this increased the infiltration of inflammatory monocytes. So this could be a potential avenue for future exploration to direct a targeted immune response against melanoma brain metastasis. Now, I would like to end by expressing my gratitude to all members of the Price Lab and to our collaborators in the Bullock, Shabani, Miller and Klibanov labs, alongside the staff at UVA who helped with this project. I would also like to acknowledge the supports given by the Focus Ultrasound Foundation in funding this work and the NIHR. Thank you very much for listening.